Good morning, sir.
Good morning. Good evening, sir.
Good evening.
Graduate pharmacy aptitude test, and it is going to be held in the month of February. And before that, we are going to prepare that all the subjects, and I hope that all the students who are going to join that this coaching institute, online coaching institute, they will get 100%. They will be qualified 100%. This is my assurance to all of you. So, in short, we will discuss the type of the mode, number of the questions. It might be known to most of the students, but till if you are going to choose the correct option, and if it is a wrong, your 1 mark it will be deducted. For the every wrong answer, 1 mark it will be deducted from your score. And if you are not giving any response, if you are not going to attempt the question, then there is no plus or no minus. So the net result, that is 0. Validity, if once you qualified that GPAT, the score of that GPAT, one 160 marks means what? 40 correct questions. If you are going to solve the 40 questions accurately, then you will get the 160 marks. This is the maximum number of marks that I am going to tell you. 160, this is the maximum number of marks. If you are getting 100%, you will get qualified the GPAT. And with that aim, then ultimately you will get the success. If you are following the knowledge, then you will get the success. Second, once if you qualified the GPAT, then you will be eligible for admission in a top universities as well as in a different institute. If you are going to apply for the NIPER, then for that purpose, first you must be he has qualified one of the top exam from that pharma sector, that is known as a GPAT. Okay, so these are the benefits that why GPAT. These are the reasons for that purpose we must apply, we must approach for the GPAT. Not only for getting admission to complete your higher education, but also to update your knowledge, to update your resume. It is important. Colleagues, friends, they also arises the such question, and because of that, we go in a depression. So before starting the preparation, we end that chapter by seeing the books. Suppose if you are going to start reading the any book, if it has a number of pages, and if you are counting the pages, then it is not possible to complete problems, how to make the time management, and how to reach to the success of that GPAT. Now, I am going to tell you a very simple story. I hope that you might be surprised that sir is going to tell that story that already we have studied it in a very story. Now the time is different. Now the students, they are very smart, and we have to modify that story. Nowadays we have to work like the crow that is shown in a right side of the presentation. That he is a very smart crow. He don't want to collect the stone and put it one by one. He is collecting a single straw, and with the help of that straw he is drinking. Proper guidance from our expert teachers during that period of the lectures, your that thirst for the success, it will get fulfilled. This is one of the smart work, because when you are preparing the different subjects, there are the number of the subjects near about 10 to 11 subjects, and when a single student is facing that 10 to 11 number, if it is put forward in present of you, then you have to your thirst for the water, it will get it will get solved. You does not need to do hard work. Just you have to digest it, you have to follow it, and you will get the success. So this is about that story. Again, I want to give one more example from that our field only. Converted into that glucose, then it is possible to absorb that glucose from the small intestine. Then the absorption of glucose it takes place, and it is assimilated into the cell to get the energy. So it is a very long process. When you are taking any complex food, the conversion of that carbohydrates into the glucose, it requires the time period of 8 to 10 hours. But in the right side, you can see. So very short period of the time that is required to get the energy when the glucose it is given. But when the complex food it is given, it requires the more and more time. In the same way, so we are going to give you the ready material that is a glucose. So the time required to read it, to understand it, and then go for the number of practice questions, and then he will get the success. Yes. तर ओके काही प्रॉब्लेम आहे फास्ट होतंय का ठीक आहे okay. So this is an online coaching institute that is going to provide you the ready material, notes, flowcharts, diagrams. So your study it will become easy. You are going to do the study in a very smart work, and in a very less period of time you will get the large success. So this is the success. This is the motto that why to join the class. This is the reason behind that. Now we will see in short what are the different subjects. Generally, the subjects for the GPAT, these are classified into the two types, that is the major subjects and the minor subjects. Pharmaceutics, pharmaceutical analysis, medicinal chemistry, pharmacology, pharmacognosy, and organic chemistry. These are the major subjects. While biochemistry, inorganic chemistry, biotechnology, biopharmaceutics, and pharmaceutical jurisprudence. These are the minor subjects. Already we have discussed that if we want to get qualified, then we need to solve the 40 number of accurate questions. So this is the data about that subjects, that which are the different major subjects and the number of questions which are required to be correct. So we need to plan according to that way, and all that planning that will be made by that our teachers, that which type of the topic, how it should be prepared, which type of the topic, बरोबर. So we are going to provide you the correct planning to qualify that exam, that which topic should be focused and how the time management should be carried out according to the major and minor subjects. Again, there is a very interesting story, the race between that tortoise and the rabbit. Tortoise is very slow while the rabbit is very fast. But ultimately, what happens in the story? Tortoise gets the victory. Because what is the reason? Reason is nothing but continuity and consistency. So when the students they are going to do the study, they are having the lack of that continuity and consistency. But if you are going to join that online coaching, continuously we are going to conduct the lectures. Along with that, there are the weekly tests, and also we are going to conduct the mock tests. So you are very busy in understanding the different topics, learning the topics, and solving the different MCQ based on that your weekly study. So because of that, your continuity and consistency it will be maintained, and finally you will get the success. Now, again, I'm going to clarify that how you will get the success or what is the benefit when a single student, if he is going to study the 11 subjects and now near about the 60 number of days are available to go for that final exam. So, according to that, only 5 days are available to study for the single subject. If any student is going to do the study, so according to that calculation, only 5 days are available for the single subject. But what happens when you are going to join that our online institute? There are more than 30 plus teachers who are doing the hard works. Suppose if a single teacher is preparing one subject for that total 60 number of days, means what? 60 days of the study material it is available for one subject. So if the total 11 subjects are there, so the your study material it will be of about 660 days. So a single student he is going to do the study for that long period of time, that is the 660 days. That long period of the study materials, notes, diagram, flowcharts, MCQs, that all are available to all of you within a very short span, within a very short period of time. Why? Because we are working in a team. Because a different expert peoples from the different expert subjects, they are available to guide you. They are doing their study. They are making the notes. They are preparing the charts, diagrams, so that your study it will become easy. You will understand it and you will get the success. Now, one of the important way is pattern of that GPAT. So you will understand it if you are going to solve the previous year's question papers. So again, it is not possible for that single students to solve that all the previous year question papers. So we will work in a team. We are going to differentiate the questions in that previous year according to the subjects. Different questions from that subject of physics, different questions from the subjects of that microbiology or from the subject of analysis likewise. And each subject teacher, each expert teacher is going to discuss that all the questions which has been asked in the previous exams. So your all that question papers they are also going to be solved during that online coaching. So this is also one of the important tasks because it is one of the major criteria. If you are solving all the question papers, then your success, your way to the success, it becomes very easy. You are going to very close to get qualified in that GPAT exams. So this is about that. This is about our planning. So I am requesting all of you that if you have not registered, so please join our coaching institute and do the registration. Once you are registered, then you will get access for that all the material, all the study material or the online test also. So these are the some type of the charts that I am going to present you. Now we are not going to discuss about that charts. But now I am going to discuss the some topics. I'm going to ask the some questions to test your knowledge. One of the first question, what is mean by glidant? So you can comment about that glidant. What is mean by glidant? Sir, are you getting the voice? Is there any answer? I hope there is no comment. Yes, still now there is no answer from a single student. Okay, glidant, these are the flow promoters which are used in the manufacturing of tablets. There are the number of the excipients, and the glidant, it is one of the excipient. It is a flow promoter that helps to promote the flowability of powder in the manufacturing of that tablets. Again, I have a second question that is from the different subjects, that which type of the chemical material it is present in the cell wall of fungi. Okay, I'm going to provide you the option: peptidoglycan, chitin, cellulose, or polysaccharide. Which type of the chemical constituent is present in the cell wall of fungi? Okay, answer, that is the chitin. Chitin, it is a type of chemical constituent that is present in the cell wall of fungi. So in short, now we are going to discuss about that cell. There are the different type of the two different type of the cells, prokaryotic and eukaryotic. Cell wall, it is present in a prokaryotic organisms, and it contains the peptidoglycan chemical material. While in a eukaryotic organism, cell wall, it may or may not be present. If it is fungi, it contains the chitin type of chemical constituent. If it is a protista, then it may or may not contain the cell wall. If protista, these are plant like, then cellulose type of the chemical constituent are present. And if the protist, these are animal like, not contain the any type of chemical material. Now again, I have a second question. Can you describe the composition? Can you tell me the different type of the blood cells? I hope that all of you are getting my voice. Okay, till now we have seen that why to go for the GPAT, and the second, that why to go for that online GPAT coaching class, why to join our institute. So we have discussed that all the benefits regarding to that. We have also, I have also discussed the planning, that how we have designed our plan for the next 60 number of the days, and how it will help you to qualify that GPAT. If you are following that our plan day to day, then 100% you will get the success. All the students, they will get the success. Now I am going to discuss the some topics, some questions from that different subjects. So my first question, again I am going to revise, that what is mean by glidant. So if you know the answer, you can put your answer into that comment box. What is mean by glidant? Okay, so okay, okay. Glidants, these are, there is no comment. Okay. So glidants, these are the flow promoters, and these are used in the manufacturing of tablet to improve the flowability of powder. So this is the one of the benefit. Yes, which improve the flow property. Yes, one of the student, he has given the answer. Okay. So the second question, again from the subject of that microbiology, that fungi, it contains the which type of the chemical constituent in its cell wall. What type of the chemical constituent is present in the cell wall of the fungi? Okay, I am going to provide the options. Option A, cellulose. Option B, peptidoglycan. Option C, chitin. Option D, polysaccharides. So the answer, that is a chitin. Chitin, it is a yes. Tejasvi, she has given the answer. So chitin, it is a type of chemical constituent which is present in the cell wall of fungi. So the prokaryotes, they also contains the cell wall. So which type of the chemical constituent present in the cell wall of that prokaryotes? Again, there is a second question. Options are same. Prokaryotic organisms, they contain which type of the chemical constituent in their cell wall? Options are peptidoglycan, chitin. Okay, if you are going to put down the answer like a chitin, cellulose, then it will be easy. Okay, peptidoglycan, chitin, cellulose, or polysaccharides. What is the answer? At least type the three initial letters of that answer. Chitin, CHI, cellulose, CL, peptido, PEP. Okay, peptidoglycan. Answer, that is the peptidoglycan. Peptidoglycan, it is a type of chemical constituent which is present in the cell wall of that prokaryotes. So these are the very basic things that you have learned early from that your first, second years. But such type of the questions, they are frequently asked into that GPAT, or in a different competitive exams also. So you need to prepare for that. Cell, it is classified into two types, prokaryotes and eukaryotes. Prokaryotes, they contain the peptidoglycan. In eukaryotes, there are the four different types: protists, fungi, plantae, and animalia. In animalia, cell wall, it is absent. In fungi, cell wall contains the chitin. In plants, cell wall, it contains the cellulose. While in case of that protist, cell wall, it may or may not be present. If it is present, it contains the cellulose type of the chemical constituent. So this is about that cell. So can you tell me bacteria? It comes under which type of the cell? Bacteria. whether it is prokaryotes or eukaryotes. Bacteria. Bacteria, whether it is a prokaryotes or a eukaryotes. Please comment your answer, whether it may be right or wrong. It doesn't matter. Okay, bacteria, it is a prokaryotic type of cell. Okay. What is about that amoeba? What is about the Plasmodium falciparum? Plasmodium falciparum, amoeba. So they comes under that protozoa, and protozoa it comes under that of the protist. Okay, no. Plasmodium, these are the protozoa. Okay, and protozoa they comes under that protist. These are the protist type of the cell. Now again we will shift to the different topic, that is the composition of the blood. The composition of the blood, it can be studied again in a single chart. If that single chart is provided to you, that chart it will contain all the different types of the blood cells, the composition of that blood, and the function of each and every type of the blood cell. So there are the three different type of the basic blood cell. All of you might be known: RBC, WBC, and platelets. So can you tell me the synonym for RBC? RBC, these are also known as, yes, anyone? The RBC, these are also known as erythrocytes. So what is about WBC? WBC are also known as leukocytes. WBC are also known as leukocytes, and platelets, these are also known as thrombocytes. So can you tell me the different subtypes of that WBC? Different subtypes of WBC. Subtypes of WBC. I'm waiting for your response. Mr. Kulvinder, if my pronunciation is right, I am waiting for your response. What are the subtypes of that WBC? Yes, monocytes. Yes. Again, what is another type? What is another subtype? So the WBC, these are basically classified into the two major types: granulocytes and agranulocytes. Granulocytes and agranulocytes. In granulocytes, there are the three different types of the cells: neutrophils, basophils, and eosinophils. While in agranulocytes, there are the two different types, that is the monocytes and lymphocytes. Yes, monocytes and lymphocytes. Again, the lymphocytes, there are the two types of the lymphocytes, that is the T lymphocytes and beta cells, B lymphocytes. So these are the two different subtypes of the lymphocytes. So again, another question is there: which type of the cells are phagocytic in nature? Which type of the cells are phagocytic in nature? Yes, students are giving the response. I'm happy to see that. I'm waiting for your response. Which type of the cells are phagocytic in nature? Mature monocytes, which are known as a macrophages. Mature monocytes are called as a macrophages, and macrophages are phagocytic in nature. So this is about that blood in short. Then what is the role of platelets? Platelets, these are the blood clotting cells. They helps to clot the blood whenever if there is a cut and the damage. So this is regarding to that cell, blood cells. Now again, I am going to ask you regarding to that vaccine. Do you know the polio vaccine? Can you tell me the different types of that polio vaccine? What are the two types of that polio vaccine? Vaccine, it is very serious issue nowadays. All of us are waiting for the vaccine. So there are the two types of the polio vaccine: vaccine and Sabin. Salk and Sabin. These are the two different names of the scientists: Albert Sabin and John Salk. John Salk, he was the scientist who discovered that vaccine, and it is an injectable one. Okay, Salk, it is a injectable polio vaccine, while Sabin, it is a oral polio vaccine. Again, the second difference, whatever that regarding to that Salk, it is a it is prepared from the killed viruses, killed microorganisms, while that of the Sabin, it is prepared from that of the attenuated. So in this way, we have to go for the preparation. In India, the Sabin type of the oral polio vaccine, it is preferred to administer to the children to get the possibility of that polio, to get the infection from that polio disease. So what is meant by vaccine? We are talking about the vaccine, types of the vaccine. Can you tell me that the vaccines, what do you mean by vaccines? A very simple definition. Please note down your comments. Okay, vaccines are defined as a live, attenuated, or a killed preparation of microorganism. Again, ask a different question from that different topic regarding what do you mean by that primary standard substance. What is meant by primary standard substance? Okay. Those the substance which are having very high purity, high accuracy, and yes, Kulvinder, he has given the answer regarding to that vaccine. Okay. So again, I am waiting for that primary standard substance. If you know, please comment your answer. It's okay. The primary standard substances are those substances which are having very high accuracy, high purity, and their solutions it is prepared as per the procedure that is provided into that IP. The given quantity, that is weigh and when quantity of that sample, it is dissolved into the solvents like water or some other solvent, and you will get the standard solution. You does not need to perform the standardization of such type of the solution, while the secondary standard substances are those which are of having inferior quality, and once if their solutions are prepared, then their standardization should be carried out. So this is the difference between that primary standard and secondary standard substances. So again, I have one more question, that the types of waters, do you know the types of water? Okay, we have to prepare that all the topics in this way. We have to go for the more and more MCQ. We have to look at the each topic in that way, in that sense, that what different types of the questions can be arise for that each and every topic. So when you are going to study, it becomes very difficult, because what is your first attempt? In the first attempt, you are going to read the books. You are reading the more and more books, and you get confused, but you does not get the proper time to solve the MCQ. If you are solving the more and more MCQ, and if you are preparing that particular type of the subject, that particular type of the topic, in that sense, then it will become very easy, and your study it will become interesting. It will become more funny if you are doing the study in this fashion. So we have to go in a next upcoming days. We have to do the preparation in the same way, that we will prepare the topic in that sense, that that topic it will be revised in the form of MCQ. Just wise that we have studied now the blood cells, and we have studied the blood cells in the way, in the format of that multiple choice questions, that how different type of the questions can be arise. We also studied the type of the cells in the same way. Again, what are the different examples? Fungi, it comes under that kingdom. Yes, fungi. Then protozoa, they comes under the kingdom Protistee. Then your plants, they comes under the kingdom Plantae. Animals, they comes under the kingdom Animalia, and the bacteria, they comes under the kingdom Monera. Again, I have one question. What is about viruses? What is about their classification? Kiran Raut, he has given the answer: hard and soft water. Okay, I am also expecting for your answers. Types of waters which are used in parenterals. Types of the waters which are used in parental preparations. I am waiting for your answer. Okay, Kulvinder, he has given the answer fungi. So WFI, SWFI means sterile water for injection, bacteriostatic water for injection. These are the types. So you must be known about that all the topics, all the type of the subjects in this way, in this sense. Then what is the difference between that of the WFI and SWFI? Again, I have one more question. That what is mean by pyrogens? What is mean by pyrogens? Yes, yes. Students are giving the responses. Pawan, Wi-Fi, WFI. Okay. So one more question. Pyrogens. What is mean by pyrogens? Pyrogens, these are the products of the metabolism of the bacteria. The bacteria, if these are present into the preparations, then they will carry out the metabolism, and due to that their metabolism, so some toxic metabolic products, these are released outside of the bacterial cell, which are known as a pyrogens. So to check the presence of pyrogen into the parenteral products, we need to perform the pyrogen test. One more test is there, that is the sterility test. Which type of the products they are exposed for the sterility test? I have the one more question, that which type of the products are subjected for the sterility test? I am going to provide you the option. First, ophthalmic products. Second, injectable products. Third, oral preparations. And fourth, option A and B both. Again, yes, yes, yes. Kiran, he has given the answer. Okay, again, I am going to ask the question that what was the question? Yes, which type of the products are subjected for the sterility test? The option A, ophthalmic products. Option B, injectable products. Option C, oral preparations. Option D, both A and B. So what will be your answer? This is a very simple question. So the ophthalmic preparations and the injectable preparations, parenteral. But what is meant by parenteral? Whether ophthalmic product they comes under the parenteral? Yes, Priyanka, she has given the answer both. Yes, ophthalmic and injectable, both type of the products they are subjected for the sterility test. Can you tell me that which type of the different culture media are used into the sterility test? Again, I have one more question, that which type of the culture media are used into the sterility test? Yes, Kulvinder. The answers: fluid thioglycolate media, soybean casein digest media, and alternative thioglycolate media. These are the type of the culture media which are used into the sterility test. So in this way, you have to go for the preparation. In this sense, you have to learn. So it will become interesting. I am going to share again the screen. I hope now you might have got the key of that success. What is the key of the success? The key of success, it is nothing but once if you are going to take the proper guidance from the proper expert team, then you can unlock the way of that GPAT. Once you have unlocked the way of that GPAT, once you got the success, then definitely your future it will become very bright. So I want to quote one here. तू खुद की खोज में निकल, तू किस लिए हताश है? तू खुद की खोज में निकल, तू किस लिए हताश है, तू चल तेरे वजूद की समय को भी तलाश है. If you are doing the proper management, if you are taking the proper guidance, if you are having the consistency and continuity, then definitely you will get qualified the GPAT 2021. These are the two things that you have to follow. So again, I am requesting to all of you to go through our schedule in detail, you go through the syllabus, and you do your registration as early as possible, so that we will be that type of the straw to make your hard work into the smart work. So learn to use the straw, try to do the smart work, so your success that is in your hand. I hope that again second, our expert teacher, he is going to conduct the another lecture, and I am going to hand over my charge to that expert teacher. Sir, can you getting my voice? uh, can you sorry, there is one little adjustment. There is one little correction that our next proposed lecture that will be held on tomorrow. So again, I am going to inform that our second lecture it is going to be held on tomorrow. Okay, enjoy your learning and get the success. Don't get bothered. Don't get tense. Learn with a fun. Okay. Good night.